Good to see you all. I'm very pleased to be here. I'm the CEO of Cereno Scientific. We are a company that is involved in developing a first-in-class prevention drug for the major killer in the planet, thrombosis, blodproppar in Swedish. And I have the great news to tell you. This morning we released a press release that we got a patent approved on the largest pharmaceutical market in the world, the US market. And thrombose prevention therapy is around $10 billion in sales every year. So that's a key milestone for us to get that patent approved. But we'll leave that and I'll get back to the story of Cereno and what we are about. So thrombosis is, you probably have family or friends that have experienced a heart infarction or stroke or even a blood clot in the veins and so on. So it's very common and it's the #1 killer on the planet. The sales around the world is around $20 billion yearly. Not only those who suffer a thrombosis in the heart or the brain or elsewhere are suffering from this disease. There are 10 times more people that survive than actually are killed by thrombosis. Every second there's somebody getting a thrombosis. Now, current drugs are used to weaken the coagulation system in the body. We have the coagulation system to stop bleeding, so that's a good system. And the blood thinning drugs weaken that system to prevent thrombosis. And that's what we have today on the market. And the problem with that therapy is that because it weakens the coagulation system, it puts everybody treated on an easy to bleed state. So there is a risk of bleeding, severe bleedings in the brain or in the stomach or elsewhere. And not only severe bleedings, but also any kind of bleed. So around 3% of those who get this therapy get severe bleeds, and up to 25% of those treated get any kind of bleed. So bleeding is an issue, and cardiologists know about this and are worried about it, but they need to treat thrombosis. But risk patients don't get therapy. Those are at increased risk of bleed. And because of this, the therapies are developed in moderate dosages, so they don't have the optimal effect that you would like them to have. And that's why one treated patients in 5 years get a new infarct or new thrombosis event. So that's 20% in 5 years that get a new event. So there is a problem in the market of bleeding, the risk of bleeding, and would like to have more preventive effect, more efficacy on the market. So, and that's exactly what Cereno is addressing. So what's used on the market? Let's look at this axis. You see it's like a line there. It's a balance line. And the body often works like that. So one system, the coagulation system, is working to heal a wound. And the fibrinolytic system is actually taking care of blood clots so they are not growing too much and becomes an occluding blood clot in the vessel. And they are normally in balance. But if they're out of balance, we are at higher risk of developing a blood clot. And what you can see on this line, to the left, there is a number of therapies used for preventing thrombosis. And they're all working to weaken the coagulation process, thus the bleeding risk. Now, Cereno is working with the body's own defense mechanism against blood clot. We are working to restore our own defense mechanism. We have such a system. So we have, if you think about this room, and there are sprinklers for fires, and they start to sprinkle water when there is a fire in the room. That's how the body is defending itself against blood clots. the body has sprinklers, so to speak, in the vessels everywhere. And if there's a small wound in the vessel, it will sprinkle out a fibrinolytic dissolving substance called TPA, and thus stop that blood clot from becoming too big and occluding the vessels. Cereno is working to strengthen that defense mechanism in humans. And let's look at how that can be illustrated. So this is a vessel that's cut in half. So we look into the vessel, and when we get older, we get atherosclerosis and we get plaques in our vessels, and they're a bit stiff. And when they crack, there is a bleeding. And so it starts to bleed, and then there are signals that go up to the wall of the vessel, the endothelial cell wall, and there sits the sprinklers. And there is a substance there that will be released by this bleed. And that's the fibrinolytic substance, the defense mechanism. And it will make sure that the clot is not too big. There is another protein in the blood floating around called pi, and that is supposed to take care of TPA when it's done its work, or if there's too much coming out of the sprinkler. So this is the normal defense mechanism. Now, the problem in risk individuals for thrombosis is that system is weakened. It doesn't work as it's supposed to be. And that is the case if you are hypertensive, high blood pressure, if you have diabetes, if you're overweight, obese, and so on. So those individuals have a higher risk of developing blood clots. And one reason is that their system of defense is weakened. And if you look at the left side here, that's illustrated by a heart. And the coronary vessels are supposed to give the heart blood and oxygen and so on. In the normal circumstance, if you have a plaque that bursts, you see there's a small blood clot forming, but then TPA is sprinkled out and take care of dissolving the clot so it doesn't become too big. And pi, the blocker of TPA, is in low concentration, illustrated by small letters. If you look to the right, there is very little TPA coming out of the sprinkler because there isn't TPA in the wall. There is too little stored by this person. And in addition, the blocking substance of TPA is too high concentrations. And you might know this, that in the morning hours, it's most common to get myocardial infarction, also stroke. And that's also when pi, that substance, the protein, the blocker, is high in concentrations in the blood. Now, what is Cereno doing about this? Well, we are working, let's jump back. Don't know. Here I should be able, sorry. So we are working, and we have found that our substance, we call it CS1, our drug candidate, can reverse the situation from the right here to a healthy and much stronger defense mechanism in the body. We will, if you take our substance in advance, that will improve the synthesis of TPA, so it's stored up in the cell wall, so it's ready there if there's a bleed. And it will also, if you have this therapy, reduce pi, so it's not in high concentrations. Yeah? The beauty of this system, and it's why it's first in class, nobody else has done this. And by pre-treating, we are not weakening the coagulation system. We are not causing the patients and putting them at risk of bleeding. We are just pre-treating to restore the proper defense mechanism of the body. So it's not our agent that is dissolving the blood clot. It's the body's TPA that's doing it. And we have made sure that it's there in the sprinkler. So we're not putting patients at risk of bleeding, and we're having a positive effect. And the body activates its own sprinkler when needed, by itself, not done by us, so to speak, or our substance. So we are moving ahead, documenting our substance, and our aim is to document the preventive effect against blood clots, and as a first-in-class, we will do this. And it will be a valuable alternative to what's out there on the market, and everybody is looking for an effective drug that doesn't cause bleed. Now, let's look at some documentation. So this is from an aorta in a mice, and they've been treated with the controlled substance and with our active substance, VPA. And the green there is TPA, the water in the sprinkler, so to speak, the protein in the sprinkler. And you can see if they're treated with our substance, they have much more TPA. So they are ready to take care of blood clot. This is in live animals. This is in mice. And they have been treated for 3 days or 96 hours. And the graph to the left shows those who have been treated and those who were controlled. The treated is the red line. And these mice were hurt, so there was a bleed in the vessel in the stomach, and there's a blood clot forming. And as you can see, the red line is much lower than the black line, and that's documenting that it's preventing fibrogen buildup, or the blood clot buildup. And already after 2 minutes, you can see they separate. So there is a preventing effect of blood clots appearing in the mice, in live mice. And to the right, you see a photograph of the thrombosis formation. So we have documented that this works in live animals. We've also been in man and documented the release of TPA and the reduction of pi by 50%, the protein floating around in the body, which is a main risk factor for myocardial infarction. Well, do we have any other evidence that this might work in man? Yes. In 2011, a Danish research group published a study on the whole Danish population. And they looked at what epileptic drugs were used over a 10-year period, and they looked at cardiovascular incidence. Were there difference between different treatment groups? Yes, in one group there was a difference. And so this is a major study, 150,000 patients. And in one group, the group treated with a substance called valproic acid, that's our substance that we are using in our development, there was a 40% reduction of myocardial infarction in this epileptic population. So it looks like this is preventing heart infarctions in man. There has been other studies. An Irish study came 2014 in 20,000 patients, same data. And actually this year there were 3 studies published with similar data, preventing effect in stroke this time. So we have developed this substance, valproic acid, into a specific innovative controlled release formulation that is targeted to take care of the pi, the floating protein in the body, that is a risk factor, and also to make sure that the TPA is synthesized and stored in the vessel wall. And we have brought this substance into man in a phase 1 trial, and we published the data in June, and it showed good safety and tolerability as expected, because it has been in man for 45 years, but not used in this patient population. We also looked for if this formulation also reduced pi. We wanted to show, because it's a specific formulation to take care of pi in the morning hours. And it did. So we have a significant reduction of pi. Very positive data for us. So we are developing into phase 2. We are in phase 2 now. And when you want to develop pharmaceuticals, you need to finance it. And phase 2 trials are expensive. So we financed the company with up to 106 million Swedish in March this year. We published the agreement. And it's a specific agreement where we can pull down funds as we need them. We did this financing agreement because we needed to secure the funds over a long period of time. It has hurt our stock. But the risk is reduced for being able to get the work done. And when we have a phase 2 study done, I'm sure that people will be happy with the valuation of the company. So New first-in-class drug, innovative formulation, short-term to market, has a double mechanism of action. Increase TPA synthesis and reduce pi. That's what we are about. So we have not said that much about our development program, more than the latest press release this week, that we have signed an agreement with the CRO. It's OCT based in St. Petersburg, Eastern Europe, and offices also in the US. We have done a procurement for over 6 months with a number of CROs, and we've done feasibility where it's best to do this trial and so on. And we ended up signing the agreement with OCT. And we will do the study in 20 centers approximately in Russia and Bulgaria. And we aim to submit the documents for approval for the study by the end of this year. What else? This is our development line. I should also mention that when gearing up for phase 2 trials, you need to increase your capacity in different ways. So we just announced this morning that we have assigned a new chief medical officer in Björn Dahlöf, associate professor from Gothenburg, Sahlgrenska University. Very well published and experienced professor from Gothenburg. He's also a major shareholder in the company and sits on the board. He now goes in operatively to work with this trial, amongst other things. He's been involved in many trials globally. We also then today released that we have gotten our patent approved, or notice of approval for the US market. So this has been a very good week for Cereno. And I hope you understand the impact that this drug will have if we succeed. I've been fortunate to be in development processes a couple of times that have changed medication globally and improved survival. And I think this drug has a possibility to do something similar. All right, thank you.
Thank you, Sten. Very interesting to hear. A huge indication you're working with, huge market, of course. Would you say, what are the biggest challenges you're up for?
Yes, I mean, if you have great ideas, there's lots of challenges. You need to be able to tell the story, and you need to create followers that believe in your story for different reasons, for competence, capacity, for financing, and so on. And that's a long road. As I mentioned, I've been doing this a couple of times before in major pharma. And even inside a company with lots of billions, it's a road that you need to travel. So I think in our situation, there's always a scientific proof and documentation that you need to do. And we have so much competence. People involved in the company were hired and developed thrombosis medication, prevention medication for AstraZeneca that are global sellers today. So we know what we're doing, we think. Yeah. So we actually think that the financing is the major challenge for this program. And as I mentioned, that's why we took this financing, despite that it also have some negative impact short term for us. It's not the only financing we will do based on our strategy, but we needed to do it. So
it's a means to an end.
Yes.
You mean? Yeah. Speaking of talent and competence, at the Nordic Life Science Days conference, industry conference that was the other day, that was a recurring subject, I think, to the difficulty it's the biggest challenge in Nordic life science companies, basically, to attract talent and to find the right competence. However, as you mentioned, you announced today that you have a new CMO. And you didn't have to go that far to find him, did you?
No, I mean, and that's actually fortunate for us. I've been working most of my life in cardiovascular medicine, and here or abroad, but globally. And fortunately, AstraZeneca, which I used to work for, my first job in '84 was for Astra, but I came back in '95 and worked globally. But AstraZeneca is one of the leaders in the world in research and has been since the '60s. So we have lots of talent to pull from. So that's fortunate. And we actually moved our company into BioVenture Hub, which AstraZeneca has opened up for smaller companies that they think have a great future and promise, and not necessarily conflicting with what they're doing. So we have talent that we can bring in. You always want to bring in, when you develop drugs or you're on this path, you'd like to bring in the best people in the world. You need them, and you can do that in phases. But because Astra has been so good at developing drugs and has been exactly in this field, we can bring the best in from a local perspective, so to speak.
Well, that's convenient. And you mentioned yourself the financing perspective, another challenge, of course. You have a solution in place now. You're looking at next step of development, and you're going into a clinical phase 2 trial, and looking to do that later this year. And that adds on, you're going to another level then in clinical phases, and there's costs related to that. Are you financially set to go into that phase, or?
Yeah, we are. And again, that's why we did the agreement we did. So the whole procurement process has involved actually defining almost down to the bare bones the details of the study we will do. So that's all done, except for the parameters in the work that needs authorities' approvals, and there's some work left there. But the whole agreement with the CRO is based on a very detailed plan, both in objectives, activities, and contents and measurements and so on, and patient recruitment times and all of that. So it's time and events based. And I should maybe mention that we are doing the study will be done in orthopedic surgery. It's a great model for the pharmaceutical industry and has been in the past to prove proof of concept in man to prevent thrombosis. And so you replace your knee. And there are 15,000 knee replacements every year in Sweden, for example. So that's a model that we will be using in measuring this.
And in terms of the next clinical phase, then what patient numbers are you looking at to include there?
We haven't released that.
Okay.
But
you can tell me.
Yeah, I am not allowed due to the stock market regulations. But it will be, there are 20 centers. So it will be multiples of the hundreds. So it's going to be it's a trial that the big pharma would do. Yeah.
Okay. And then going forward, it's going to require, providing everything goes as planned, you're going to need to do even bigger studies, next coming phases. And the strategy there is to find a partner who can help you do that, or?
Well, somebody said, I was at a CEO forum for listed companies within this field, and somebody said that who'd been very successful in making a deal recently, a major multi-billion deal in Swedish kroner this summer, and said that their philosophy was that we'll always tell the world that you'll do it yourself as long as you can, and then whoever is interested to come and be partner with you, they're welcome to discuss. And I think that's our philosophy. We'll move with this concept until the world understands how good it is. But normally after phase 2, if you have very good data, that is a valuation inflection point for the valuation of the company. Normally, a project or a company If you pass phase 2, you have proof of concept in man normally. And that's when big pharma or partners want to go in, because they want to form the phase 3 program themselves. But we might pursue a phase 3 ourselves too. So we haven't, we're open.
Okay. We're looking forward to learning more about that in time. Thank you very much for coming in today.
Thank you.
Yeah.
Thank you.